Now I will be discussing about the RNA polymerase types. So before going ahead with the types of the RNA polymerase, we should be having an idea about what is RNA polymerase. RNA polymerase is an enzyme. So what are enzymes? They are made up of the polypeptide chains. It is basically a protein. Now this RNA polymerase plays a very crucial role in the central dogma. So in the central dogma, what happens? In the central dogma, we all know that the DNA is expressing itself and then forming the RNA, and then that RNA makes the protein. So now the genes of the DNA that is expressed are read by the RNA polymerase. This RNA polymerase reads the genes of the DNA and then forms the RNA. Now, in the case of the formation of RNA, there are two types of cells, that is prokaryotes and eukaryotes. In them, there are different types of RNAP, that is RNA polymerase, are present. We will be going ahead with the discussion of them. Now, we will be discussing RNA polymerase types in eukaryotes. So, there are 5 different types of RNA polymerase. So there are 5 different types of RNA polymerase. In that, there are 3 major types of RNA polymerase, that is RNA polymerase 1, and then we have RNA polymerase 2, RNA polymerase 3. These are the 3 main RNA polymerases. Now, how were they discovered? They were discovered by a toxin which is present in a mushroom, that is called as the alpha amanitin. So this toxin is very sensitive. This toxin is very sensitive to the 2 types of RNA polymerase, that is RNA polymerase 2 and then RNA polymerase 3. So the RNA polymerase 2 and 3 are inhibited by this toxin, that is alpha amanitin, but it does not have any impact on the RNA polymerase 1. Okay, so on this basis they discovered the 3 types of RNA polymerases. Now we will be proceeding ahead all the RNA polymerases individually. So there is a mnemonic called as, that is roommate. So this mnemonic helps us to remember the different types of the RNA polymerases. What do they produce? So if we take a look, so the row here in this row, it codes for the RNA polymerase 1. So in the second one, that is MA, it is coding for the RNA polymerase 2, and then this TE, it is coding for the RNA polymerase 3. Now if we look it carefully, the RNA polymerase 1, as here R is present, it forms the rRNA. Now here there are 2 types, that is zeros are present. By this you can just think it as a nucleoli. So the speciality of RNA polymerase 1 is it is only present in the nucleoli. Okay, nucleoplasm of a cell, it is only present in that. Now when we take the look at the RNA polymerase 2, the RNA polymerase 2 synthesizes the mRNA. So here mRNA you can take from the M. Now it is also inhibited by a toxin. So this A stands for the alpha amanitin. So this alpha amanitin inhibits the RNA polymerase 2. Now next we have this third one, that is the RNA polymerase 3 synthesizes tRNA. So again there is a E present here. So this E is, as it is in the 5th letter of the alphabet, it codes for the 5S RNA. Okay, so now here we can look at it carefully. This is the rRNA, which is produced from the RNA polymerase 1, it forms 45, that is sRNA. This 45 sRNA, it again ruptures to form 3 more RNA, that is 28 sRNA, and then we have 18 sRNA and then 5 point sRNA. These are the precursors of the rRNA. So by this we have got a knowledge of the what are the functions of the RNA polymerases with the mnemonics. So there is another one, that is the RNA polymerase 2. As we look at it here, if we break this RNA polymerase, it will be forming a small fragments. By this we can conclude that the RNA polymerase also forms the small RNA, that is microRNA, and then that is small RNA or sRNA are produced from this RNA polymerase 2. So now we will be discussing about the RNA polymerase in prokaryotes. RNA polymerase in prokaryotes, as we looked at the RNA polymerase in eukaryotes, there were 3 different, that is majorly 5 different were there. In that we discussed about the RNA polymerase 1, 2, 3. But in case of the RNA polymerase in prokaryotes, there is only one, that is RNA polymerase, that is called as the RNA polymerase holoenzyme. So this RNA polymerase holoenzyme, it codes for the rRNA and tRNA and then mRNA. It codes for these 3 things. Now if we look at the RNA polymerase holoenzyme in the prokaryotes, it is a multi-complex enzyme. Multi-complex means what? Multi-complex means here it is made up of many subunits. Now if you look at the RNA holoenzyme, it is made up of some of the types, that is core enzyme and the sigma factor. So the core enzyme includes here, that is the alpha subunit, there are 2 types of it, and then beta subunit, beta dash subunit and omega subunit. So there is another, that is sigma factor. So this RNA holoenzyme, that is RNA polymerase holoenzyme is there, no? It is inhibited by a antibiotic called as the rifampicin. So as we have known that whenever we get the bacterial infection, so doctor give us this solution. So now if we go ahead with the functions of these core enzymes, the alpha subunit, it binds to the promoter region. Next we have the beta subunit, it acts as the catalytic sites and binds to the NTP, and then it also helps in that is giving the energy to the RNAP. Now we have the beta dash subunit. This beta dash subunit acts generally bind to this RNA holoenzyme, and then it gives the strength and then facilitate the movement of this RNAP holoenzyme. Next we have the omega subunit. It helps in the assembly of RNA polymerase. Now comes the role of the sigma factor. The sigma factor is recognition of the promoter region. So it helps in the recognition of promoter region. Okay, these are the functions of the RNA polymerase holoenzyme.